Hello and welcome. It's now 4 o'clock, so we should get started. So let me just close down these little windows. Welcome to this webinar. I'm Ann Holmberg from Qlucore, and I will talk to you today about statistics, both new and existing, as well as the new Biomarker Workbench. So I hope it will be quite exciting. So let's get started. So the agenda, I've changed my screen, so I take away the video again. The agenda, we will start with the built-in statistical methods, the standard methods, so I will go through all of them and highlight the changes and new things. And then we will have a look at the Biomarker Workbench, which is a really exciting new functionality, and we will end with how to work with the extended statistics, which is not anything new. It's in there, but many of you have maybe not used it so far. So the statistical tests that are available in the software is the Ttest, which we call the two-group comparison. And we would today also talk about the paired Ttest and the new two-way analysis. Then we have the Ftest, or the multi-group comparison, and regression analysis, both linear regression as well as rank regression, we'll show you. And then we also have some R-scripted tests like Welch, Lima, Mann-Whitney, and Kruskal-Wallis that we will have a look at how to use. So I'm actually just jumping in here, not giving you any additional background to Qlucore and so on, but we just jump right in. And I will focus a lot also on the actual tests, not so much on the complete workflows and not so much on interesting data sets, but this will be more focused on the actual statistics today. And then we have a lot of other webinars if you would like to get more aspects of the software. But today we focus on the statistics. So we start with the two-group test and see what we can do there. So here we have the software. I will load the example data set, and we actually changed that a bit. We added more samples to it and more annotations, as you can see here. So we can use it for more cases. And we want to color this according to treatment. So we have 3 treatment groups, and you can see that the color scheme has also changed from what you are used to. And now we actually give different annotations, different colors automatically. So if you're not happy with the colors, you can either just double click in the color box and change it to something that you like. If you want to go back to the old color scheme, you can do that by just opening the more plot settings and go back to old scheme. So that's still available if you prefer that. Okay, so here we are. We have 3 treatments, and now we want to do a t-test. So then we open the statistics dialog, and you notice that these up here, we have now icons here that are new. So to open the statistics dialog, if it's not already open, you click here. So then I pick the test that I want to do, a t-test on treatment. And the old way of doing this is what we see here. I pick one of the groups, and then the other group is the rest of the samples. So in this case, the combination of drug 1 and drug 2. So in this setup, I test placebo versus the combination of drug 1 and drug 2. And I can drag the statistical slider to find my genes of interest. Maybe that was too tough. So So with the Q value, a false discovery rate of 0.14, I find 7 variables, 7 genes. The Q value of 0.11, I find 3, and so on. And I can inspect the result here and see that the placebo group, there's a fairly good separation between the placebo samples and the treated samples. Not fantastic, but fairly good. Okay, and of course I have the result here in the variables pane, and I can create a copy of that list, Ttest on placebo versus treated or something like that. Okay, but now we also have a new option. So if I would like to compare placebo with drug 2, I can now pick another group here. And the meaning of this now is that the statistical test is only performed on the samples belonging to placebo and drug 2. So the genes, the variables that I find, they are good at separating these two groups. But the visualization includes all samples. So sometimes you are interested in doing this and then see how that affects all of the samples. If this is confusing, you always have the option, as you did before, to take away the group that you are not interested in, and then you only visualize. Then the visualization and the statistical test, they are matching. But you see, it doesn't, if we look here, the Q value doesn't change, and the number of variables doesn't change when I take this away, because the test is already restricted to only handle these two groups. So now it's actually the same if you choose placebo versus all or placebo versus drug 2, because drug 1 is not active. So this is a bit different from what you are used to. So if it's confusing, just take away the samples that you are not interested in. Otherwise, this is a type of something called restriction. So we are restricting the test to only placebo and drug 2. So that was the first thing. So let's talk a little bit more about restrictions. So here we are. So now we will change to an Ftest, a multi-group comparison. And an Ftest, we use that when we want to separate all the groups in an annotation. And in this case, we have 3 groups, 1 placebo and 2 treated groups here. So when I do the test now, I'm looking to find genes or variables that are good at separating the 3 groups from each other, as you can see like this. But again, I have the option to add a restriction. So if I add a restriction here, for instance, saying that I only want to work to find variables that are good at separating the treatment groups for the females. So now I'm finding variables that are only looking at the female samples, but I'm still displaying all the samples here. So again, if I would like maybe to highlight which are the female samples, I can use mark up here and click on females, and then they are marked. So I see which are the females and which are the males. And maybe from this I can see that it's easier, that these variables are better at separating the female samples than the male samples. And again, if I want the visualization to match the test that I did, then I just take away the male samples and then I only see the female samples like this. So that is how restriction works. And you can decide if you want to use it or not, but it's there, it's available. Okay, then we move on. And if you have any questions, just use the chat and I'll try to answer. And now we move on to the two-way analysis, which is a new test that we have not had before. So it's now available here. Sorry, let's talk a little bit about what this is. So a two-way analysis, with that you can test the combination effect of two sample annotations. So in this example here, I want to see if the different treatments, if they are different between the males and females. So let's see how that works. So then I go back here. Let's take away the mark like that. And then we go over here to the two-way analysis. And then I pick the two annotations that I want to combine. You could do this before as well, but then you need to create this combination annotation. But now you have support for it here. So two-way analysis, treatment, and I want to combine it. I choose to combine it with time point instead. So there are two time points here, pre-treatment and post-treatment. So now I want to see, is there a difference between the treatments before and after treatment? And you see here that it looks like it, because here we have drug 1, drug 2, and drug 1, but only half of the samples. So let's pull up a second PCA plot here to understand what's going on. And let's color this on time point. And let's change the color to something else here. It's easier to see like that. So here we have, or maybe it should be a totally different color, not the same as we've been using in the other plot. I changed it to yellow instead, sorry. So here we have the post-treatment, so after treatment, and it's clear that I don't think this is a synchronized plot, so I do this again, new synchronized plot. I want the plots to be synchronized. So then I again color this on. So now you see, so when the plots are synchronized, of course, everything that I do affects both plots at the same time. So that's very often what I want to do. Here you see the samples after treatment. And here you see the samples before treatment. And yeah, it's obvious, of course, that the patients that have received the treatment, well, after the treatment, there we have a clear effect, while before treatment you don't really have an effect. So this is how you do a two-way analysis. You can easily combine annotations. So you have three different output modes. You have the combination output mode here, where we want to combine and see what we have. We also have the output mode treatment, and this means that I'm testing treatment and taking away the effect of time point, so eliminating the effect of time point. And the output mode B, meaning that I'm testing time point and eliminating the effect of treatment. So this is a new test that you have available. So let's move on, and I will send out these slides to you after the webinar as usual. Now we will have a look at the paired T-test, which has been possible to do before and is still possible. So then we have paired data, and that's typically when you have one single experimental factor and each subject receives all level of the factor. You have 2 samples per subject. For instance, a study where each patient is studied before and after treatment. And another example is when you have 2 sets of tissue samples from both tumorous tissue and non-tumorous tissue from each patient. And in both these cases, the patient acts as his own control. And this type of data is sometimes referred to as paired data. So now we are going to work with another data set where this is very clearly demonstrated, this effect. So this is a public data set with microRNA expression profiles associated with colon cancer. So here we have 85 patients, 170 samples, and 2 samples from each subject, one from tumor tissue and one from non-tumorous tissue. And let's have a look at that. So we close down this data set like that, and we open up the example data set. So here we are. And let's start by, let's start by coloring this data set on tumor like this. And here we can see that we don't really see any clear difference between tumor and non-tumor. We see something else, but I will come back to that. So there is no clear difference between tumor and non-tumor as far as we can see. So let's do a T-test and see if we can find such a difference. So I do a T-test on tumor versus normal. So I can either pick normal here or just pick all. There are only 2 groups in here, so it doesn't really matter. And then we try to, so we filter down to a Q value around 0.01, and then we find 24 microRNAs. And we can see here that they are, they are fairly good at separating the samples. There's quite a mixed zone here as well. But now if I add the eliminated factor, I make this a paired t-test. I do that by adding the eliminated factor patient, like that. And now I use the multi-group test here. Before we only had 3 options, now we have more options here. So I picked the multi-group test because we have 85 patient groups containing 2 samples each. So this is the correct setting. And now when I did that, you saw that. So now I remove the effect of the individual patient and I focus on only the difference between tumor and normal. And that made the picture much clearer, as you can see. So now I have found 174 microRNAs here that are distinctly different between tumor and non-tumor. So when you have the possibility to work with paired data, it's really good because working with real patient data is a challenge because there are big differences between the normal levels of individual patients. So being able to use the patient as its own control really makes it easier to analyze this type of data, as you can see. Yeah, so that was the way that you can set up a paired t-test. So let's continue here. And so when we talk about eliminated factor, another word for that is covariate. A covariate is a variable that is not controlled in the experiment, but may still affect the dependent variables, the ones that we are interested in. And typical examples of covariates are age, sex, and batch effects. And in Qlucore, the covariates are represented by sample annotations. So let's have a look at another example of this, and we can continue with this data set. So let's reset this and remove this eliminator factor for the moment. So this is where we started, and we could actually see that it looks like the data is divided into 3 groups. And we also see that there seemed to be an equal distribution of the tumor and non-tumor within these groups. So they are not explained by the annotation tumor. They are not explained by the annotation patient, of course. But we have 3 batches. Let's see, color by that. And then we can see that, yes, the annotation batch actually seems to explain these 3 groups. And we already saw that the tumor and non-tumor samples were equally distributed between the batches. So that means that it's probably safe to eliminate this factor. So then we can eliminate this effect by doing again, a multi-group analysis or choosing the multi-group analysis alternative for the annotation batch. And this matches the old option where we said nominal. And then you see that here we now take away the effect of batch. So we center around the mean value for each of these groups, and they are now all centered around the middle of these plots. And then we can continue with our analysis. So if I now also, then I just can continue and add the patient. So do my pair Ttest, and let's color on tumor again. And here I now find actually a few more variables than before, like that. No big difference in the outcome, but still. And you know, of course, if I continue and show a little bit workflow here, then I can have a look at my results here. I pull up a synchronized plot. I might, as a heat map, I might want to hierarchically cluster. I might want to color on my annotation of interest like this. If I'm not happy with the hierarchical clustering, I would like to have a clearer picture. You can always force the ordering of the heat map. Say I want to order on tumor instead. Then it's of course not unbiased. It's biased because you have decided how you want to sort, but it makes the differences between the groups show clearer. And then, of course, if you want to zoom in, you will eventually also see the, in this case, the microRNA descriptions here, like that. Then if you want to go back and see everything again, you can do that. And if you want to export a plot, you just right-click here, export image, and pick one of the file formats here and export like that. That goes for all the plots, easily exported. And if you want to save a list, you just go to the active list, make a copy, rename it, and then you can export it from here as a TXT file. So it's very easy to also save your results like this. So that was just a quick little workflow showing you what more you can do once you have found your variables of interest. So, okay, And I just got a question here. Can I also exclude a whole list of variables from my data set? Yes, you can do that, but then what you need to do then is, so let's just go back and show everything here. I can kill that plot for the moment. So then if I would like to exclude these, my best genes, for instance, here, maybe not that probable, but then, so I've saved a list of that. If I would like to exclude these genes, the way I do that is to use the set operations tool and say, make the difference between my full data set and this list called the copy, this one, and apply. That creates a new data set. And now I have a new data set with only 1084 genes. And then I can use that as an input and continue to work only with that. So now you see that I only have 1084 active variables. If I would like to save this data set, then I would have to go here, File, Export, Data, and then I would get the reduced data set. The question, do I want to export all variables or only the active variables? And then you export that, and then you have a new data set, a G data set of only these 1084. Yes. So that was the long answer to that question. Okay, so now we continue and go back to the more statistical tests. So then we close this one down and open the test data set again. That. And now I want to do a linear regression, and I use that when I have numerical data. So let's see. So we have the annotation age here, for instance. So let's have a look at that one. Here. And so if I color by that, you see that doesn't really help me. I don't understand what the colors mean. So maybe I would like to create a color gradient. Then I use this icon here. And then I want to color arrange as gradient. This is the option I want. Then select the first value in the range and press OK. Yeah, let's do that. So that's the first value and that's the last value. And the values must be in the correct order. So now I have a color range from gray to green. Maybe that was a bit hard to see. So I can also say that I would like to start on yellow. Okay, and I would like to end on black. Black. Okay, and then I do this again. So I start at yellow and I want to end on black instead. Then it's slightly easier to see. So now yellow represents a low age and black represents a high age. Now I do the test, linear regression, pick my annotation, age, and then I drag the statistical slider here to see what I can find. So I can find two variables with a Q value of less than 0.05. I go back. The next one has a Q value of 0.19. Maybe I'm okay with that, maybe not, like that. So we see here that I have it. So we have a clear tendency of lower ages here and higher ages here. So we have found something. And maybe I would like to show this now in a scatter plot. So let's pull up a scatter sample plot like that. That comes up empty. I must define the axis. So I want the sample annotation age on the x-axis, and I want a numerical scale. And here I pick the second option. I want the variable selected by the y-axis tool, which is the mouse. Then I go to the variables tab and pick my active list, and then I get my two variables here, as you see. If I want to see only one of them, I pick that one. And I can also, if I want color on something else, maybe treatment, for instance, then I get some information there. So this is useful when you have a truly numerical annotation. It only works if you only have numbers. But sometimes you also have maybe ranked values. So in this case, we have 3 age groups, and the description here is not purely numerical. It could also be low, medium, high, or something like that. And then we use rank regression. So here we have rank regression like that. And let's see here, go back to the PCA plot and change here to age group like that. And here it's important that the groups are sorted in the right order. As if they are not, you can use the arrows to move this up or down like that. So that's very important because we are not using the actual values here. We're just using the order. So I pick rank regression, I pick age group, and then again I drag the statistical slider to see what I can find. Like this. So I can find not fantastic with a Q value of 0.19, the best I can do. I see that we have a reasonable separation. The blue ones are the lowest ages, the gray ones in the middle are the middle group, and the yellow ones are the highest ages. So some kind of ranking here as well. Yep, and then we also support quadratic regression. Okay, so now I'm going to show you a little bit on the new feature Biomarker Workbench, which is really, really nice. Here we can run several tests in batch mode, so we can easily find where we have the most interesting information in our data set. And we can also explore correlation between sample annotations. So let's see how that works. So then we open the Biomarker Workbench here. Here we are. Let's maximize it. And before we start with that, there is some new terminology. We talk about explanatory variables. So that's a combination of statistical tests and sample annotations, as you are used to. But now we changed the name here to explanatory variables. And we also have something called response variables. And these are both the traditional variables like genes, proteins, and so on, but now also sample annotations when we are looking for correlation between sample annotations. So let's see how this is done. So here we are. And now I can add explanatory variables here. So for instance, I'm interested in treatment. a multi-group test on treatment. I'm also interested in time point, a Ttest on time point. I'm also interested in age, linear regression on age. And you can actually add all your annotations if you like. Then I would like to eliminate sex, because I'm not interested in differences between male and female here. So I eliminate that factor. And then I just press run. And then I get the result. So here I get an overview. And you can see that I find 7 genes with a Q value lower than 0.05 for treatment, 4 for time point, and none for age, with the eliminated factor of sex. And then I have the details here. So I can add and remove columns. So I add gene symbol and I take away the p-value columns, so we don't have so much information here. So here we are. So here's the minimum Q value. So that's any variable where we have a good response in any of the tests. So here you can see that here they are. If I want to focus on the treatment, I click on that to sort on that column. And then you can see that I found, I think, 12 genes here with a Q value lower than 0.1. If I go over here, I see that I have for treatment, I find 4 with a Q value, with a good Q value, and for age, I don't find any at all with this setup. Okay, and then if I would like to let me think about the order. Yes, I think it's the first time I do this webinar, so but maybe I would then like to export a list of the variables that are best for separating the treatment. Then I just mark this variable and then I can create a variable list, best, so Ftest treatment, like that. Ftest, like that. Okay, like that. And then if I go over here, you will find that that list is now here, like that. And if I now make this list the input, let's color on treatment first so we can see the effect. So if I make this list input, you see that these 12 genes are indeed good at separating the treatment groups. So there is a seamless interface between the Biomarker Workbench and the rest of the software. Okay, so let's now have a look at response variables, how that works. So then I take away age for the moment, and then I want to focus only on the response variables, and I add all the annotations as response variables. So then I take away the ones that we are comparing with. This is the eliminated factor. Okay, and it suggests a test that matches the data. So if it's linear, it suggests a linear regression. If it's numerical, it suggests a linear regression, and so on. And you can, of course, change that. And then I just press run. So now I'm looking for correlation between these response variables and the annotations I have, the explanatory variables that I have defined up here, like that. And here I have the results. So you see that here I can see that I have a correlation between patient ID, maybe not that interesting, also with alive or not alive, and also more interesting with blood pressure. So blood pressure could be a factor that I should have a closer look at here, actually. So here you see in a very short time you can find interesting correlations here between your annotations, like that. So let's see, next slide. Yeah. And then we have something called controlled experiment. So let's just kill these for the moment here, like that. So if I now click controlled experiment here, and before I do that, I add, let's say, age here again, like that. If I click control experiment here, that means that I now test each of these annotations and at the same time eliminate the effect of the other ones. So when I click run here now, you see that okay. So this is something that you need to be aware of. When you make a list and input like this, then those are the only active variables. So then when you want to continue with your test, you need to untick that box so that all your variables are active again. So that's why I didn't really find anything here. So let's press, so then I must make a change here again to activate this. So I want to add the age again, like that, and press run. And yeah, actually didn't find anything. So then we see that for each test here, we have taken away, here are the eliminated factors. So here we have eliminated time point, age, and also sex. Here we have eliminated treatment, age, and sex, and so on. And then if I find interesting results here, I mark the test that I'm interested in to continue with and copy to statistics. So now you see that this test is now here in the statistical dialog. So the multi-group comparison on treatment and then all the other added as eliminated factors. So again, it's very easy once you have found something interesting here to just continue with the analysis. Now I realize why I didn't find anything. It's because I said none there, which is a good setting when you do response variables. But here, when you want to test this, of course, you want to have all your variables active. So that's why this happened. Good. So I think this is an extremely interesting piece of functionality that you should jump in and start playing with your data set. So especially if you have a lot of annotations in your data set, clinical annotations, for instance, it can be a very tedious work to explore all of them manually. So with this, the Biomarker Workbench, you really have the possibility to try this out or to quickly identify interesting signals in your data set. Yep. Okay, so then I will move on to the last piece of this webinar. So no questions so far on the Biomarker Workbench? No? Then we'll continue. So in the extended statistics, you can run R scripts. So you can either add your own R scripts if you are fluent in R and like to R script your methods, or if any of your colleagues produce R scripted methods, you can add them to Qlucore. And there are also some additional tests included in the software, like Welch, Mann-Whitney, Kruskal-Wallis, and Limma. And these, the Mann-Whitney and the Kruskal-Wallis, are non-parametric tests. And the prerequisites for this is that R has to be installed on the computer where you run the Qlucore software. and you must activate the extended statistics mode. I will show you how to do that. And if you write your own R scripts, you must do it according to a specified template, which is described in our reference manual. Yeah, so let's have a look at how this works. So I close down the Biomarker Workbench and we go here. So extended mode, this is the tab that you want to see. So if you don't have it active, you can activate it from File, Preferences. I think the preferences are located a little bit differently on the Mac, but I'm sure you will find it. And here we have the statistics. So the extended statistics mode must be activated. And this is the folder where the R scripts are stored. So, and this is where you should also put your own R script if you write R scripts of your own. And if you do that, you should also change the folder to somewhere else so that you don't overwrite your own R scripts when you update the software. Okay, and how does this work? And we go to extended mode, and then we pick the test that we want to do. So we have two group tests like Mann-Whitney and Welsh, and we have multi-group tests like Kruskal-Wallis, Limma, and a special test here for Limma and Womb if you have count data, unnormalized count. Then we do the normalization and Limma in one go. Okay, but let's say I want to run Kruskal-Wallis on treatment, like that. Then I must press run, because here we are running R scripts, so it's not automatic as you are used to. So you must press run, and then all the P and Q values are calculated as normal. And then you can apply the P value, the statistical slider, like that. But if you change anything, like for instance, I don't want to include drug 1, then you need to press run again. So every time you make a change, you must press run. And R is slow. Qlucore is much faster than R, as you will see if you run this on bigger data sets. Yeah, so that's so here you have access to additional methods if you're interested. And if you want more information on how to write the R script, you go to the reference manual, you search for R script, and here you have it. So here you can find out the syntax for how to write the R scripts. Great, so I think that was all I wanted to tell you today. So do you have any questions or comments? Use the chat if so. While you are thinking about that, let me tell you a little bit on upcoming webinars. Tomorrow we will talk about the new genome browser, which includes ChIP-seq and ATAC-seq analysis. So if you already have the NGS module, or if you're interested in the topic, please join that. And next week we are doing a couple of webinars, go through a lot of different plots that you can produce, the basic hands-on training if you're a new user, and a more specific webinar on how you can import and analyze public data from the SRA archive from GEO and TCGA. And we continue to run more webinars on different themes. So by that, if there are no questions, I would like to thank you so much for your time today and wish you good luck when you try all this new functionality. And if you have any questions, just email me or my colleagues Jan or Jana if you're already in contact with them, or support, of course. So thank you so much for your time today, and I look forward to hearing from you. And have a great rest of the day. Bye-bye. And if you have time to fill in the little questionnaire at the end, that's very much appreciated. We all love to get feedback from you. Thank you. Bye-bye.